For thousands of years, across diverse cultures and continents, our ancestors understood a profound truth about human vitality. They didn't rely on synthetic compounds or complex pharmaceuticals. Instead, they harnessed the power of the earth itself, utilizing specific plants to stay young, strong, and remarkably disease-free. This wasn't anecdotal folklore, it was lived experience, passed down through generations. Today, modern science, with its advanced tools and rigorous methodologies, is finally catching up, providing irrefutable evidence. We now confirm what these ancient civilizations always knew: certain botanicals contain compounds so powerful they can actively reverse cellular aging, rebuild damaged DNA, and demonstrably add healthy years to your life. And the most astonishing part? Most people in the modern world have never heard of them. Our investigative journey begins with ashwagandha, Withania somnifera, a cornerstone of Ayurvedic medicine for over 3 millennia. Often dubbed the Indian ginseng, its applications range from boosting energy to promoting tranquility. But the true scientific interest lies in its profound impact on stress and, crucially, cellular longevity. Chronic stress, as numerous studies confirm, is a major accelerant of the aging process, eroding telomeres and fueling systemic inflammation. Ashwagandha intervenes directly here, demonstrating its potential to not just manage stress but to actively mitigate its aging effects at a biological level. Central to ashwagandha's anti-aging prowess is its documented ability to modulate cortisol, the body's primary stress hormone. Elevated cortisol levels, sustained over time, are a known biomarker for accelerated aging, linked to everything from immune suppression to cognitive decline. A landmark study published in the Indian Journal of Psychological Medicine in 2012 examined the effects of ashwagandha extract on individuals with chronic stress. The results were compelling, indicating a significant and measurable reduction in circulating cortisol levels among participants receiving the herb. This direct impact on a key aging pathway highlights ashwagandha's sophisticated biochemical intervention. The numerical data underscores the efficacy. In that same randomized, double-blind, placebo-controlled clinical trial, participants who received a high-concentration full-spectrum ashwagandha root extract demonstrated a substantial reduction in serum cortisol levels. This wasn't a marginal shift, it represented a statistically significant decrease compared to the placebo group. Such a significant attenuation of cortisol indicates not just symptomatic relief from stress, but a direct biological intervention that could protect against the cellular damage stress inflicts, thereby slowing a key driver of intrinsic aging. Beyond cortisol, emerging research points to ashwagandha's influence on telomeres, the protective caps at the ends of our chromosomes. Telomere shortening is a recognized hallmark of cellular aging, directly correlating with lifespan and disease susceptibility. A study published in PLOS I in 2015 investigated compounds capable of influencing telomerase activity, the enzyme responsible for maintaining telomere length. While direct human trials on ashwagandha's telomere impact are still evolving, in vitro and preliminary animal studies suggest its adaptogenic compounds may promote telomere stability, offering a fascinating avenue for direct anti-aging intervention at the genetic level. While human data is nascent, preclinical studies provide intriguing insights. Research in specific cellular models and animal studies, investigating compounds similar to those in ashwagandha, has shown a potential to significantly influence telomere length. For example, some compounds have been observed to increase telomerase activity, leading to a measurable elongation of telomeres. This indicates that ashwagandha's adaptogenic properties might extend beyond stress reduction, directly impacting the genetic markers of aging. This frontier of research, while still early, offers a glimpse into direct cellular rejuvenation pathways that could one day be clinically validated in humans. Beyond stress and telomeres, ashwagandha demonstrates neuroprotective properties crucial for cognitive longevity. Studies in animal models, such as those published in the Journal of Ethnopharmacology, have shown that ashwagandha extract can promote neurogenesis, the growth of new brain cells, particularly in the hippocampus, a region vital for memory and learning. This ability to rebuild and repair neuronal networks directly counters age-related cognitive decline, positioning ashwagandha not just as an anti-stress agent, but as a brain-rejuvenating botanical. This complex interplay of effects highlights its holistic influence on the aging process. Next, we turn our attention to turmeric, curcuma longa, the vibrant golden spice ubiquitous in Southeast Asian cuisine and traditional medicine. Its primary active compound, curcumin, has been the subject of over 15,000 peer-reviewed scientific articles, drawing intense scrutiny for its profound anti-inflammatory and antioxidant capabilities. Chronic inflammation, often termed inflammaging, is a silent orchestrator of age-related diseases, from cardiovascular issues to neurodegeneration. Turmeric, through curcumin, offers a potent defense mechanism, directly addressing one of the fundamental drivers of accelerated biological aging. Curcumin's molecular mechanisms for combating aging are multifaceted. Research, notably in journals like Antioxidants and Redox Signaling, demonstrates its ability to scavenge free radicals, preventing oxidative stress that damages cellular components and DNA. Furthermore, it modulates various inflammatory pathways, including NF-kappa B, a protein complex that controls DNA transcription and is central to inflammatory responses. By downregulating these pathways, curcumin not only reduces existing inflammation but also protects cells from future damage, fostering an environment conducive to cellular repair and regeneration, a critical component of reversing the aging process. A critical aspect of cellular aging involves the decline of mitochondrial function. Mitochondria are the powerhouses of our cells, producing ATP, the energy currency of life. As we age, mitochondrial efficiency decreases, leading to lower energy production and increased oxidative damage. Studies, including those cited in molecular nutrition and food research, indicate that curcumin can enhance mitochondrial biogenesis, the creation of new mitochondria, and improve their overall function. This boost in cellular energy production is not merely about feeling more energetic, it's about providing the fundamental fuel for all cellular repair and anti-aging processes. Quantifying curcumin's impact on mitochondrial health reveals significant effects. Research published in PLOS One demonstrated that curcumin treatment in various cell models could lead to a substantial increase in mitochondrial respiratory capacity and overall biogenesis markers. In one specific in vitro experiment, a key marker of mitochondrial density and activity showed an impressive uptick. This isn't just theory, it's a measurable enhancement of the fundamental energy systems within our cells, directly supporting the body's ability to combat age-related decline and maintain youthful cellular vigor. Similar to ashwagandha, curcumin has shown promise in promoting neurogenesis. A study in stem cell research and therapy explored curcumin's role in boosting levels of brain-derived neurotrophic factor, or BDNF. BDNF is a crucial protein that supports the survival of existing neurons and encourages the growth and differentiation of new neurons and synapses. By increasing BDNF, curcumin helps maintain brain plasticity, a key factor in cognitive longevity. This mechanism not only protects against neurodegenerative diseases but actively contributes to a more resilient and youthful brain, even as chronological age advances. Despite its myriad benefits, curcumin faces a significant challenge: poor bioavailability. When consumed alone, it is rapidly metabolized and excreted, meaning very little reaches the bloodstream or target tissues. This discrepancy between lab potential and real-world efficacy has led researchers to investigate methods for enhancement. One of the most effective solutions identified involves combining curcumin with piperine, the active compound in black pepper. Piperine has been shown to inhibit certain enzymes that metabolize curcumin, dramatically increasing its absorption and retention within the body, ensuring its anti-aging power can be fully realized. The difference in bioavailability when combining curcumin with piperine is stark, transforming its therapeutic potential. Without piperine, a significant portion of ingested curcumin is simply rendered ineffective, passing through the system with minimal impact. However, the addition of even a small amount of piperine, as documented in a study from Planta Medica, can drastically alter this dynamic. This synergistic relationship highlights the importance of formulation in harnessing the full anti-aging benefits of botanicals and underscores why traditional practices often combine multiple ingredients, demonstrating an intuitive understanding of complex biochemical interactions. Perhaps one of curcumin's most compelling anti-aging applications lies in its potential role against Alzheimer's disease. The accumulation of amyloid beta plaques in the brain is a hallmark of this neurodegenerative condition. Research, particularly in preclinical models, such as that published in the Journal of Alzheimer's Disease, has investigated curcumin's ability to cross the blood-brain barrier and interfere with plaque formation. While human trials are ongoing and complex, the preliminary findings suggest that curcumin's anti-inflammatory and antioxidant properties contribute to a neuroprotective effect, offering a glimmer of hope in the fight against age-related cognitive decline. In various animal models of Alzheimer's disease, curcumin has demonstrated a remarkable capacity to reduce amyloid plaque burden. A study detailed in the Journal of Neuroscience found that curcumin supplementation significantly decreased the total amount of soluble and insoluble amyloid beta in the brains of mice models. This reduction translates to a direct amelioration of one of the primary pathological features of Alzheimer's. While not a cure, this evidence suggests a powerful preventive or supportive role for curcumin in maintaining brain health, underscoring its potential as a formidable anti-aging agent for cognitive function. Our third plant, green tea, Camellia sinensis, is a beverage consumed for millennia, particularly in Asian cultures renowned for their longevity. More than just a comforting drink, green tea is a powerhouse of polyphenol, most notably epigallocatechin gallate, or EGCG. EGCG is a potent antioxidant and anti-inflammatory compound, far surpassing many commonly recognized superfoods. Its role in combating oxidative stress and systemic inflammation makes it a foundational component of any strategy aimed at reversing cellular aging and promoting overall vitality. The scientific community has extensively validated its broad spectrum of health benefits. One of EGCG's most significant contributions to anti-aging is its ability to activate autophagy. Autophagy, meaning self-eating, is a fundamental cellular process where the body cleans out damaged cells, proteins, and organelles, recycling them to make new, healthier cells. It's essentially the cell's sophisticated waste disposal and recycling system. As we age, autophagy declines, leading to an accumulation of cellular junk and dysfunction. Research published in Autophagy indicates that EGCG can significantly stimulate this process, effectively performing cellular housekeeping that rejuvenates cells from within, a direct mechanism for reversing cellular aging. Green tea's EGCG also shows a direct link to telomere maintenance, another critical aspect of cellular longevity. Studies, including a human intervention trial in the American Journal of Clinical Nutrition, have investigated the association between green tea consumption and telomere length. While the direct causal mechanism is complex, research suggests EGCG may upregulate telomerase activity, the enzyme responsible for adding repetitive nucleotide sequences to the ends of chromosomes. This activity is crucial for preventing telomere attrition, directly protecting our genetic material from degradation that underlies aging and age-related disease, a powerful anti-aging effect. Quantifying the impact, studies have revealed that regular consumption of green tea catechins, including EGCG, can positively influence telomerase activity. A human study involving individuals consuming green tea regularly showed a measurable increase in telomerase activity markers compared to non-consumers. While specific percentage increases vary by study design and population, some in vitro models indicate that EGCG can boost telomerase activity by a significant factor. This direct enhancement of the enzyme that rebuilds our genetic clocks offers a profound biological pathway to slow and potentially reverse aspects of cellular aging, pushing the boundaries of longevity science. Beyond cellular repair, green tea's catechins contribute significantly to metabolic health, an often overlooked aspect of anti-aging. Dysfunctional metabolism is a key factor in age-related diseases like type 2 diabetes and obesity, which in turn accelerate biological aging. EGCG has been shown to enhance fat oxidation and improve insulin sensitivity. Research published in the American Journal of Clinical Nutrition demonstrates these effects, indicating that green tea consumption can optimize energy utilization and help maintain a healthier body composition. By supporting a youthful metabolism, green tea acts as a systemic anti-aging agent, reducing the burden on cellular systems. The metabolic effects of green tea are quantifiable. A meta-analysis published in the International Journal of Obesity reviewed numerous studies and consistently found that EGCG supplementation significantly increased fat oxidation, particularly during exercise. In specific trials, participants showed a measurable increase in fat utilization as an energy source, rather than carbohydrates. This enhancement of metabolic flexibility is critical for maintaining healthy body weight and reducing the risk of metabolic disorders that accelerate aging. This data confirms green tea's role in promoting a more youthful and efficient metabolic profile. Finally, EGCG's neuroprotective actions contribute to cognitive longevity, a vital aspect of healthy aging. The antioxidants in green tea protect brain cells from oxidative damage, while its anti-inflammatory properties reduce neuroinflammation, a contributing factor to cognitive decline. Moreover, EGCG can modulate brainwave activity, promoting alertness and focus without the jitters associated with excessive caffeine. This holistic support for brain health, documented in reviews in phytomedicine, means green tea doesn't just keep your cells young, it helps keep your mind sharp and resilient, directly addressing the mental aspects of aging. Our fourth powerhouse is Rhodiola rosea, often called golden root, an adaptogen native to the Arctic regions of Siberia and northern Europe. For centuries, it has been revered for its ability to enhance physical and mental endurance, reduce fatigue, and increase resilience to stress. Like ashwagandha, rhodiola's adaptogenic properties mean it helps the body adapt to various stressors, but its unique active compounds, salidroside and rosavin, offer distinct anti-aging benefits, particularly in the realm of cellular energy and mitochondrial health. It's an ancient secret for modern vitality, now rigorously validated by science. The key anti-aging mechanism of Rhodiola rosea lies in its direct impact on cellular energy production, primarily through the optimization of mitochondrial function. Salidroside, one of its main bioactive compounds, has been extensively studied for its ability to protect mitochondria from oxidative damage and enhance their efficiency. A review in Phytotherapy Research highlights how salidroside can stimulate ADP synthesis, the fundamental energy currency of all cells. By ensuring robust mitochondrial health and energy supply, Rhodiola helps cells maintain their repair mechanisms and youthful function, directly combating age-related energy decline. The impact on cellular energy is measurable and significant. Studies, including in vitro experiments published in Journal of Ethnopharmacology, have shown that treatment with Rhodiola extracts, particularly salidroside, can lead to a notable increase in ADP production within cells. This isn't just a marginal bump, it's a quantifiable enhancement of the very fuel cells need to function, repair, and replicate. This direct boost in energy currency means that Rhodiola is not just masking fatigue but providing the underlying biochemical support for sustained cellular vitality, a critical factor for reversing internal aging processes. Beyond the cellular level, Rhodiola's influence translates into tangible anti-aging benefits for physical and mental performance. Chronic fatigue is a pervasive symptom of aging, often linked to mitochondrial dysfunction and prolonged stress. Rhodiola, through its adaptogenic action and support for ADP production, directly addresses this. Clinical trials, for instance, in phytomedicine, have demonstrated its efficacy in reducing fatigue and improving endurance in both healthy individuals and those experiencing stress-related burnout. This means not only feeling younger but having the physiological capacity to sustain activity and mental clarity that define youth. Clinical studies have provided quantifiable evidence of rhodiola's impact on fatigue and endurance. A meta-analysis of multiple studies, often cited in reviews, shows consistent improvements. For example, a study involving individuals suffering from chronic fatigue demonstrated a significant reduction in symptoms. In terms of physical performance, athletes supplementing with rhodiola have shown measurable gains in endurance capacity. These statistics are not just about temporary boosts but reflect an underlying physiological enhancement that contributes to a more youthful and resilient state, defying the typical decline associated with chronological aging. Our final plant, ginseng, Panax ginseng, holds legendary status in Eastern medicine, its name Panax deriving from the Greek panacea, meaning all healing. Revered for thousands of years as an elixir of vitality and longevity, its primary active compounds, ginsenosides, have now captivated modern science. Ginseng is renowned for its broad spectrum of benefits, from immune support to cognitive enhancement, all contributing to its profound anti-aging profile. It's not just about treating symptoms, it's about fortifying the body and mind against the very mechanisms of aging, making it truly a root of longevity. Ginsenosides are the primary drivers behind ginseng's anti-aging effects. These saponins exhibit potent immunomodulatory, anti-inflammatory, and antioxidant properties. Crucially, research published in Journal of Ginseng Research indicates ginsenosides can influence NAD+ pathways, which are central to cellular energy production and DNA repair. NAD+ levels naturally decline with age, contributing to mitochondrial dysfunction and genomic instability. By modulating these pathways, ginseng effectively promotes cellular rejuvenation, enhancing the body's intrinsic ability to repair damage and maintain youthful function at a foundational molecular level, a direct intervention against aging. The impact of ginseng on NAD+ pathways is a significant discovery for anti-aging. While direct human stat values for overall NAD+ increase are complex, preclinical studies in oxidative medicine and cellular longevity have demonstrated that specific ginsenosides can significantly upregulate key enzymes involved in NAD+ synthesis and recycling. This translates to an enhanced capacity for cellular energy production and more efficient DNA repair mechanisms. This molecular intervention suggests ginseng is not merely masking the signs of aging, but actively re-energizing cellular processes that directly govern how quickly our cells decline, promoting genuine internal rejuvenation. A truly remarkable aspect of ginseng's anti-aging profile is its documented effect on stem cell proliferation. Stem cells are the body's repair crew, capable of differentiating into various cell types to replace damaged tissues. As we age, stem cell activity declines, impairing the body's regenerative capacity. Research, including studies in molecular and cellular toxicology, has shown that ginsenosides can stimulate the proliferation and migration of various stem cell types. This enhancement of endogenous regenerative processes offers a profound pathway to literally rebuild and rejuvenate tissues throughout the body, providing a powerful internal mechanism against aging. The numbers demonstrate the potency. In various in vitro and animal models, ginsenosides have been observed to significantly increase the proliferation rate of mesenchymal stem cells and other progenitor cells. For example, some studies, such as those published in Cellular Physiology and Biochemistry, have reported a substantial multiplication in stem cell numbers or activity markers following ginsenoside treatment. This represents a direct and measurable boost to the body's intrinsic ability to repair, replace, and rejuvenate, a cornerstone of reversing aging from the inside out and maintaining tissue integrity across the lifespan. Ginseng's benefits extend significantly to cognitive function, addressing age-related memory decline and mental fatigue. Ginsenosides enhance neurotransmitter activity, particularly acetylcholine, which is crucial for learning and memory. Clinical trials, including a meta-analysis in Cochrane database of systematic reviews, suggest that ginseng can improve memory, attention, and executive function. This cognitive boost is not merely a temporary stimulant but a sustained enhancement that supports brain health, resilience, and adaptability throughout the aging process, offering a critical defense against cognitive decline and maintaining mental sharpness well into later years. The cognitive enhancements attributed to ginseng are robustly supported by data. Human clinical trials, often using standardized memory and cognitive assessments, have shown measurable improvements in various parameters. For example, a study published in Journal of Clinical Psychopharmacology indicated that participants supplementing with ginseng demonstrated a significant improvement in working memory and reaction time. This numerical evidence underscores ginseng's capacity to not just preserve but actively enhance cognitive faculties, offering a tangible benefit for mental longevity and a sharper, more youthful mind. This is a direct intervention against one of the most feared aspects of aging. While both Panax ginseng and American ginseng, Panax quinquefolius, are revered for their health benefits, they offer subtly different anti-aging profiles due to variations in their ginsenoside ratios. Panax ginseng, or Asian ginseng, is traditionally considered more stimulating and invigorating, with research focusing on its energizing and cognitive-enhancing effects. American ginseng, conversely, is often perceived as more calming and cooling, with studies highlighting its immune-modulating and blood sugar-regulating properties. Understanding these distinctions allows for targeted application, ensuring the selection of the most appropriate panacea for specific anti-aging goals, reflecting ancient nuanced wisdom now backed by modern chemistry. One of ginseng's most fundamental anti-aging contributions is its role in promoting DNA repair and maintaining genomic stability. Damage to our DNA is a constant threat from environmental factors, metabolism, and age itself, leading to mutations and cellular dysfunction that drive aging and disease. Ginsenosides have been shown in studies, such as those in Journal of Ginseng Research, to activate various DNA repair pathways, helping cells correct errors and preserve the integrity of our genetic blueprint. This foundational protection ensures that cellular processes can continue to function optimally, directly extending the healthy lifespan of our cells. What these 5 plants, ashwagandha, turmeric, green tea, rhodiola rosea, and ginseng, collectively reveal is a powerful convergence of ancient wisdom and cutting-edge scientific validation. Their mechanisms, while distinct, often overlap in targeting the fundamental hallmarks of aging: chronic inflammation, oxidative stress, mitochondrial dysfunction, telomere attrition, and declining cellular repair. These botanicals don't offer a magic bullet, but rather a sophisticated toolkit of natural compounds that empower the body's intrinsic ability to resist, repair, and even reverse the ravages of time at a cellular and molecular level. They remind us that the solutions to profound health challenges often lie within the natural world. The implications of this research are profound. It suggests that while chronological aging is inevitable, biological aging, the rate at which our cells and systems actually degrade, is far more plastic than previously understood. By integrating these science-backed botanicals into a holistic lifestyle, encompassing proper nutrition, regular exercise, adequate sleep, and stress management, we possess a tangible capacity to influence our longevity and quality of life. The path to a healthier, more vibrant future may not be found in futuristic labs alone, but also in the ancient, often overlooked, wisdom of the plant kingdom, now illuminated by the rigorous lens of modern science.